les nouvelles provenant de cette épidémie pour voir est-ce que vraiment c'est notre type ou bien sur le génétique. Donc c'est ce qu'il essaie de voir. Est-ce qu'ils sont vraiment reliés à l'épidémie ou pas? Par exemple, I'm just going to give one example of what has been for this grade 1B, especially in the Kamitoka. Donc là, ils ont pris quoi? Vous savez, il y avait grade 1, il y avait le variant grade 1 au départ. Mais les souches d'une sous-région qu'on appelle Kamituga, aussi du Kivu, c'était aussi des Mpox. Mais quand on a pris les séquences qui étaient connues et les nouvelles de Kamituga, c'est là où on s'est rendu compte qu'ils forment un autre cluster. Mais en les mettant ensemble avec ceux qui sont connus. Donc c'est ça la question, effectivement. On prend ceux qui sont connus, on met les nouvelles, et comme ça on voit ce que ça donne. Ils sont connus, et les références.
And you see, you could also be answering different questions at different times. So let's say, for example, you're saying the clade 1B started last year, September. So you have a new sequence, and you're like, is this new or is it old? So you have a background sequence of all the different clades for monkeypox. And then you see like, okay, it's clustering with clade 1. Then you say, it's okay, it's clade 1. But then you notice that it's quite different from clade 1, right? So essentially you're answering this question, like, is it part of the same clade or it's a bit different? So you do a bit of investigation like, okay, how are the clade, the original clade 1 sequences different from each other? And how is this different now from all these other ones? Then you realize like it has more mutations than you'd expect, right? Then you say, this is probably a new clade. Right now, you know, because we are interested in the evolution of clade 1B, we can just get a tree of clade 1B. There's no need to put, you know, clade 1, clade 2A, and clade 2B. Because now what we are interested in is like understanding this specific outbreak. So like, okay, do we have, for example, things from South Kivu and North Kivu clustering differently from like what is, you know, on the west of DRC, like Kinshasa? And if we think about, you know, we have samples from Kenya, Uganda, Burundi, are these clustering differently from other, you know, from DRC sequences? So you could, depends on what you want to answer that. Okay? Now, the thing that I also need to emphasize at this point is, in your phylogenetic analysis, do not expect to build one tree. And do not expect to build the tree once. Okay?
Alors, dans la phylogénie, il aimerait insister là-dessus. Il dit qu'il ne faut pas vous attendre à générer un seul arbre phylogénétique. Mais aussi, il ne faut pas espérer à générer une seule fois l'arbre phylogénétique.
You'll do this many times because, one, you generate a tree. Then you see one sequence that has a very long branch, for example. Maybe the quality of that sequence is not good, or a couple of sequences. You remove them. You generate the tree again, right? And then maybe you say, okay, I'm not interested in this part of the tree. Because, let's say, at first we generate a tree that has clade 2A, clade 2B, clade 1A, clade 2B, clade 1B. And then you say, okay, I'm going to remove clade 2. So now I have just clade 1A and clade 1B. Then at some point, maybe you just want to generate a clade 1B sequence only. Or sometimes, even for clade 1B, you know, like there's Steven, he comes from Uganda, right? He's only interested in Uganda sequences. He does not care about sequences from other places, right? So remember that phylogenetics will be an iterative process. I don't know what is the French word for iterative. Yes. So you'll repeat again and again. So even for the bioinformatics you've done in epidemiology, do not expect that you run one workflow once and done. You need to sometimes run things multiple times. Okay? Yes.
I think what you said happened.
Yes.
Like one of the sequences, like a mistake, we did one of the sequences that was developed, and no how it was in context of people. It had like over 80% in content, and on the tree it was very long, and it was, we were wondering why we said that. But when we went back, their sequence was just worked out and we had to know the process.
Exactly.
The same thing also in our sequences now what we have one sequence which is among the Kamituga cells. Normally, when you make comparative analysis, many times each country, for Mpox, they are in the same cluster. But you have only one sequence who is among Kamituga sequences. We are wondering, is it true or is because of there are some
Yes.
mistakes in the sequencing? Now we didn't make conclusion, we are still analyzing to see whether we have to check adapters or something like that.
Exactly. You're raising very important points, and we'll talk about like what conclusions, depending on how much information you have, you can take from what you see on a tree and what you cannot. Okay? But just remember that this is an iterative process, and as you think about your samples, think about the questions that you want to ask. Because then again, we'll see that generating trees is also not a trivial process. Yeah, some things you need to run for several hours, so you want to be very sure and certain about the sequences. And also, like, you can partition your different data sets depending on questions, different questions you want to ask. All right. So we've selected our samples. You remember we said the first step in phylogenetic analysis is to create multiple sequence alignment, correct? All right. So here we are looking at an alignment of assemblies. In the bacterial world, you can create an alignment of reads. But that's a different context, okay? Here we're just going to look at alignment of assemblies. Yeah, the bacterial world is different. They call variants based on reads, which is slightly different. So you've done an alignment. I mean, we'll have a special section where we go through the small details with regard to alignment. But let me just go through the overview at this point. The other thing that we often do is also select a reference. Okay? So Peter sort of talked about this when we were doing our assembly, reference list, about the choice of the reference. How you select your reference is very critical. Okay? If you select a poor quality reference, you will also get very poor quality alignment. If you select a reference that is so distant from your samples, you'll also get to call the wrong variants that are not useful for your specific context, right? And you cannot also use one of your samples from that same outbreak as a reference, right? So the selection of the reference is very, very, very important.
Is the reference a root? If you go back to the phylogenetic tree, so you are told this is a rooted tree and one is an unrooted tree. Is the root the reference?
Ideally, yes, it should be a root, but obviously not. They should share most traits and common ancestor with your sequences. The specific word we use is an outgroup, right? So it should be an outgroup to your specific sequences. And I don't know if I have a set of slides on how to select an outgroup, but one, it should be closely related enough to your sequences, but not clustering within your sequences. And it shouldn't also be too distant, right? such that, okay, we are looking at monkeypox. Let's even say now we have also clade 1A out there, right? And we do know that for clade 1A, we have a sample from 1971. But we do know that, there are probably outbreaks that happened in the 80s, in the 90s, earlier in 2007 and things like that. We don't want to check the 1971, right? We want to check something that is recent because those mutations are more contextual to our scenario than something that happened way back in time. Okay? So, yes, definitely something that's an outgroup if you're thinking about treated tree versus unmuted. But then also, like, it should be close enough to what you're looking at. And one of the things, is that as you choose through your reference, if there's a section of your genome that is not found in the reference, it will not be aligned and be used as a foil. This is a common problem with especially insertions. Not so much for deletions, because, for deletions, typically, again, depending on the aligner, you just might split your sample into blocks, right, to align it. But for insertions, it's going to completely miss it, right? It's the same thing that often happens with, but I think NextClade does report insertions. Yeah. So again, this is just something to be aware, but it also depends on the tool you're working with, whether it does allow for insertions and deletions or it only calls mutations. Okay. All right. So using a reference genome that is close to your samples will enable you find more polymorphisms and build the phylogeny. Okay. All right. We talked about user-defined region masking, so I think probably we'll skip this because we have talked about this, but I'll just reiterate. So we want to build a phylogeny based on positions that have ancestral origin. Remember, a phylogenetic tree is a representation of the evolutionary relationships. All right? Sometimes you do have problems with repeat regions. So this might be ancestral, but it's difficult to align. Okay? And we know, because when it's difficult to align, how the alignment happens might be a bit arbitrary. So that you end up with a tree, it's showing you differences. A lot of those differences are coming from that region that are difficult to align. And it's not because those differences actually exist, it's just that it's computationally difficult for the tool to align the sequences. I don't know if you understand. So you might have a tree that maybe every time you run it, you get different results. Because remember, an alignment is a stochastic process. Stochastic versus deterministic process. Like the alignment, let's say, sometimes it can depend on, ideally, if you have 10 sequences, every time you run it, you should get the same tree. Correct? But if you have a difficult region within the alignment, sometimes you might get slightly different topology every time you run it, right? If you know you have such difficult regions that are just going to cause confusion and not give you the true differences in your alignment, then you might want to mask.
Ce que j'essaie d'expliquer, c'est que, idéalement, quand vous faites l'alignement et quand vous générez le phylogénétique, vous devez avoir le même arbre phylogénétique. Ça, c'est idéalement. Mais des fois, il y a des régions qui sont plus complexes qui font que, quand vous générez l'arbre phylogénétique, il est trop différent du premier que vous avez généré. Et c'est pour cela qu'il faut maintenant masquer ces régions qui causent les problèmes.
And often you will know this based on the pathogen. Okay? So this is where like knowing your pathogen is quite important.
Et ça, pour savoir cela, vous devez connaître un peu l'agent pathogène. C'est pour cela qu'il est essentiel de connaître l'agent pathogène sur lequel vous travaillez.
Like, I mean, I've worked with RSV and lots of respiratory viruses. We did not have to mask any region.
Par exemple, avec les virus soutien respiratoire, on n'a pas besoin de masquer les régions.
Even for SARS-CoV-2, I don't remember us like lots of workflows masking anything.
Parce qu'il n'y a pas de régions voilà, qui vont causer des problèmes de l'alignement. C'est pour ça qu'il faut connaître l'agent pathogène sur lequel vous travaillez.
Now, the other regions we often also want to mask are recombinant regions.
Une autre région qui est très importante et qu'il faut masquer, c'est surtout les régions recombinantes.
So, let me just go back here. Do we all know what recombination is? Okay, good. So, let's say we have two pathogen populations in the same person, right? Often what you expect is A will copy A, right? And B will be copied onto B. So you have eventually A and the child, and then you have B, and then you have the child, right? But sometimes biology is interesting. You have A, and you have B. Do we know what a polymerase is? Polymerase?
Alors là, il essaie de montrer que chez un patient, par exemple, vous avez deux populations d'agents pathogènes. La population A et la population B. Et après, la population A donne son petit qui est A'. La population B donne son petit qui est B'. Il y a une demande là-bas, est-ce que vous connaissez la polymérase?
The enzyme that does the copy, right?
L'ADN polymérase.
So sometimes it can copy one and then slip and then continue copying the other one.
Des fois, ça peut copier sur le premier ADN, par exemple, et descendre faire sur l'autre.
So you'll end up with A. So let's say you end up with a mix. Or maybe let's
Have A and have
yeah.
B.
So you end up with A, B, half, and B, you end up with something like that. Okay? Now, remember, a phylogenetic tree is, it shows the evolutionary history going back in time. So let's call this like vertical inheritance from the parents to the, like that, to the child going down. Now, if you think about now let's say there is a recombination between A and E. And we have a sequence F. Where do you think that sequence will be?
Donc, il on suppose que nous avons une recombinaison entre A et F et ça génère une autre séquence qui est F. Une recombinaison entre A et E. Et ça donne une nouvelle séquence qui est F. Et c'est Raoul.
It's difficult, right?
No, she said that it will be another group.
Okay, before we go into, like, okay, what proportion of the genome did you inherit from A and what proportion of the genome did you inherit from F? The whole concept about recombination violates the principle of a phylogenetic tree. Because in a phylogenetic tree, you expect that vertical inheritance of traits. Like this parent to this child to that child, like that. But now, we have where things get recombined. Even if, like, let's say, most of the genome is from A, right? And on a tree, we can place it here. But do you think it's correct? Because it's still like, let's say, 30% of that pathogen's genome is from E. Do you think you're getting to the right conclusions by placing it here? I mean, remember, a tool, when you use it, it does not care. Right? It will give you something. Phylogenetic tool, it will always give you a tree. It's the same thing like if you give a phylogenetic tool bad data. Do you think it knows? No. It will give you a tree.
Donc ce devoir ici dit que maintenant la séquence là, supposons on dit qu'elle est proche de A, n'est-ce pas? Parce qu'on sait ce qu'est la phylogénie, c'est comme un arbre vertical. Le parent donne des caractères à l'enfant, son petit-fils, petit-fils. Mais maintenant, le F là, il a pris 30% de A, 50% de F. Et on le met à côté de A. N'est-ce pas? Est-ce qu'il est réellement? Il est à côté. Mais la machine a l'outil pour faire la phylogénie. Quand vous donnez la séquence qui n'est pas bonne, est-ce qu'il va le savoir? Oui, il a juste travaillé. Donc c'est ce qui était expliqué ici.
Let me see if I can get something. How many? Okay, let's see if we can get this to work. Okay, good. I have a tree here, okay? So this was one of the early trees that was built for SARS-CoV-2, okay? Now what I want us to follow is the human sequences, okay? So this cluster is for the human sequences, right? And it clusters together with this sequence that is from a bat. Is it possible to? Let's see. Okay, the first important information, we have 4 trees. Each tree is from a different gene. Okay?
Là, on a 4 arbres phylogénétiques. Il faut savoir ça au départ. Et chaque arbre phylogénétique provient comes from
A different gene.
c'est à partir d'un gène différent.
Okay? So, in this first tree, we can see that the SARS-CoV-2 sequences are clustering with this bad sequence.
Donc, on peut voir que les séquences provenant des échantillons humains dans le même groupe qu'un échantillon provenant de la chauve-souris.
Remember, at this point, what they wanted to know was, where did SARS-CoV-2 come from? Did it come from a bat? Did it come from a pangolin? You know, what's the closest sequence that they share with the most recent common ancestor? Okay? So if you're using this tree alone, you'd say, well, probably, you know, it came from this bat, RMYN02. Okay?
Cool. Si on voit là-bas, on peut dire que ça provient de la séquence là de la chauve-souris.
Okay, then let's look at B here. When we look at B, this gene, it clusters most closely with rat G13, okay? Which is a totally different bat virus, right? Where is RMYN02? Very distant, far away, okay? Good. Now let's look at example C here. In C, it clusters closely with pangolin coronavirus, right? Okay, now we are confused. What is happening, right? Then we finish with D. So here, it's clustering closely with these two, right? But still, these two have a most recent common ancestor, and that recent common ancestor is what they share with the SARS-CoV-2 sequences. And do you know this problem is because coronaviruses in general recombine a lot. And secondly, the recombination rates is very different across different genes. So, if you have a pathogen that undergoes recombination, often you want to remove the recombination before you do a trace. Good. All right. Any questions? People are very happy, huh? C'est très clair. Oui?
How will you break a recombination? Where the recombination region?
So for pathogens that recombine, typically this is known. So one, we do have tools that check for recombination, presence or absence. In addition to that, for a lot of pathogens, in addition to just identifying, like, okay, there's a likelihood that there's recombination with this pathogen, we often do phenotypic studies in the lab, right? For example, you can mix two populations within the same species and you know that they are very different in this region. And then you do sequencing. And if you sequence, you know, the product, because of course, the pathogen will multiply, then you see that, oh, okay, for this one, I'm having bits of the genome from this one pathogen and bits of this. It's the same thing that happened with, you know, things like XBB, for example. Okay? If you're following the recombination that goes on to SARS-CoV-2. So we do have a couple of tools that check for recombination, like RDP. We can give you, I think we can look at all the computational bits tomorrow morning, you know, go over what we talked about today, Kraken, Quast, recombination testing, RDP, tomorrow, just so that we do this computational as well. And then, so we said you'll have to give like a bed file that specifies the regions that you want to mask. Then these regions will be removed from the multiple sequence alignment that is used to build the phylogenetic. Okay? Right. We're going into the phylogenetic reconstruction methods. Hang on. Good?
James, stretch.
Okay. Let's stand and stretch a bit. section.
Understand very well.
The recombination.
If you want to introduce a sample in the
Phylogenetic tree.
yes, first thing you do. That's why you do a lot of questions.
Okay, stand and stretch fast.
Let me leave the stretching part of it.
Okay, yes, service.
So you all know coconut, right? Coconut.
Coconuts.
So let us write coconut. Coconut.
Different examples. Okay? That if you're looking at an outbreak, it's different from, you select samples differently from when you're doing, like, let's say, you want to identify a novel or a new genotype. And the fourth option here is you don't have a question. You are just doing discovery, right? And in where you're doing discovery, often like you want to include all samples that you have. So you don't have any specific question. So how you select the samples will depend on what question that you want to answer. So first decide, what do I want to answer? And then select your samples. But also something we've said is phylogenetic analysis is an iterative process. You might start with no question. You include everything. And then you see interesting things, right? Then maybe you say, I'm seeing something new. Then you select a different set of samples, right? So again, the question that you answer might also, change over time. But it's important to understand like what question you want to answer and then decide on the samples. You're good? Good. Okay. So you start from a tree. You start from an alignment, you get to a tree. There are different ways of constructing phylogenetic trees. So here things will get a bit complex. Okay? On one side, we say, so let's just say on the left, simple. On the right, complicated. So you have no explicit model of evolution. Here you have an explicit model of evolution. So the way I would describe it is, if you think about what you see is what you get. Have you ever heard the phrase before? What you see is what you get. So, like often in evolution, if something changes from an A to a T, all that you care about is it changed from an A to a T. Okay? But if you go into complex evolution, you might start asking yourself, did this change from an A to a T? Or did it first change from an A to a G, then maybe a T? So that I didn't see some G in between. So you're starting to think about some models of evolution. Like, you're starting to think about the process of evolution. But on this other side, you don't care. You deal with what you can see. Right? So, one of those is the application of parsimony. Parsimony is like, who has ever heard of the Occam's razor? Which says that the simplest solution is the best. How do you say that in French? The simplest solution is the best. You don't want to, complicate things. Parsimony tree. They're usually the simplest. Like, you just look at the changes that explains, the tree, and that's it. Nothing complicated. Now, on this other side, where you have explicit model of sequence evolution, again you have two arms. So you have pairwise comparison of sequences, and then you have those that employ a bit more statistical, rigorous methods. Okay? So here we have distance-based methods, such as bio, MJ, or neighbor joining methods, or something that they also call upgamma. And on this other hand, you have either maximum likelihood or Bayesian statistics. Okay? Oops.
You gotta give it like 5 minutes.
Yes. Ah.
On fait le cours de 3 crédits en un jour.
Okay, so just know that we have different methods of phylogenetic tree reconstruction. Okay? I know you're asking yourself, which one do we use? Right? Choosing the right method or software. So I will start with on the left. Okay? We've talked about accuracy and speed, right? So here we have methods that are very fast. So these ones are typically neighbor joining and approximate maximum likelihood tree. And some of the software that you could use for these are like Mega, Pau, and Fast. Okay? And often these ones we use to give you a preliminary snapshot. Like you just want to see the initial tree. It's not the final, just the initial tree. How does it look like? Because they are very fast. And at the end, you have methods that are very slow. And the things that they do are also very complex. How many have run a beast tree before? Arnold, how long has a beast, what is the longest time a beast tree has run for you?
4 months.
4 months. Can you tell them like a beast tree can take 4 months?
I'm still gonna say this.
4 months. Your computer is running and hot. 4 months. Cut one. But you see, it's because what you understand, what you want to learn from the tree is quite complex. So it takes time. Okay? Then you have something in between that, it's a balance. It might take a day or two, or even like a couple of hours, and can still give you, good outputs about the things that you want. Now, when you run something fast, it does not mean that it's always wrong. I want you to understand that. And it's the same thing like if you use the wrong data sets, even if it takes months, it can still be wrong. Okay? Again, going back to the fact that we have an iterative process. That, sometimes you get your new data, you just want to see how things look like. So you might run like approximate maximum likelihood tree. Nowadays, even something like IQ tree can give you a very good tree for maximum likelihood, and it's also pretty fast. Okay? So bioinformatics methods are also improving as we go along, because people are learning how to calculate these parameters quickly, but also more accurate. Okay? So, so in addition to the speed, you also think about the complexity of the data. Okay? Because obviously, if you're analyzing a whole genome, vis-a-vis analyzing a small proportion of the gene, it's going to be different. Maybe even for a gene, even for beast, it might take even 1 hour, right? Because it's not complicated, right? But for some pathogens, even a single gene can take you days, because it's just, there's too much variation, and it's trying to understand the data. Remember also, the other thing you have to consider is the concept of time. If, like, let's say, we are looking at the outbreak samples between September and December. Remember we said that as the pathogen spreads, it accumulates mutations, right? So the data gets a bit more complex, correct? So the time frame that you're also looking at will determine the complexity of the data. All right. So we had 2 arms. Remember, no explicit model of evolution. Here, we are looking at some model of evolution. We are trying to explain the evolutionary process. So, as part of tree building, we have different tree building models. Okay? So, these models allow for the correction of genetic distances for multiple substitutions or unseen evolution. Okay? So let's look at this figure here. So we have, for example, let's talk about the nucleotide substitution models. We have A, T, G, and C, right? Which are the purines and which are the pyrimidines? A and T are purines, right? Or pyrimidines? Pyrimidines. And these ones?
No, purines are A and G.
A and G are
Purines.
purines.
This is
Now this is where people don't know basic biology. Which ones have similar structure?
Purines in A and G.
Okay.
Yes.
Purines are A and G.
Yes.
T and C are
Pyrimidines.
pyrimidines, right? Okay. So when you think about substitutions, so okay, I think we'll have to teach people biology 101.
Yes.
So anyway, let's think about mutations, right? An A can change to a G, right? An A can change to a T. An A can change to a C. But an A can also change to an A.
Yeah.
Because in the copying process, it can copy it correctly. So that's also a possible substitution. Correct? All right. So in these models of evolution, we start asking ourselves, what is the probability of A changing to a G? What is the probability of A changing to a C, of an A changing to a T, and A changing to A? Then you start thinking about, those concepts of purines and pyrimidines, right? These two have the same structure, right? these two have the same structure. So you can argue that there is a higher probability of A changing to a G than an A changing to a T, correct? Just thinking about the physical structure. Because if you remember, I think this had 3 bonds, this had 2 bonds, right? So just thinking about the structure. Then you could say like, okay, I'm going to give higher probability for changes between A and G and T and C. And then, for example, you say that I'm going to assign equal probability of an A changing to a G and a G changing to an A because they are of the same class. Okay? Other than the probability of change, we also have to think about frequency. You can assume that in my genome, A, T, G, and C have the same frequency. So each of them is what percent? 25, right? Because 100 divided by 4 is 25. Or you can say, okay, I think my genome is GC rich. Okay? So I'm going to say, I'll assume that the frequencies of A and G is 60%. And T and C is 40%. Right? So these are assumptions. And remember, modeling is based on assumptions, right? We are trying to model these frequencies and probabilities. Because we are saying that we don't just want to believe what we are seeing. There is something that probably we did not see. Okay? And in fact, for some phylogenetic analysis, remember you can give your alignment that has ends, right? Some tools will say, okay, any position that has an end, I will throw it away. Okay? There are some tools that will say, okay, I do not know what N is, and I'm going to assume that it's a 5th nucleotide. And for such a position, I'm going to add an additional complexity, N, and I will also calculate the relative frequency and probabilities, including N. Okay? So, we have simple to complex models. The simplest one is what we call the Jukes counter. So Jukes counter, for example, here assume that the base frequencies are equal. Each of them is 25%. And the transition probabilities, which sometimes they call exchangeability parameters, are also equal. Like the probability of a change from an A to a T is the same as A to a G and G to a C, it does not matter. Okay? Then you have, for example, the Filson-Stein 81. So it still assumes that, you know, the transition probabilities are the same, but the base frequencies are not. They are free to change wherever. Okay? So, then there is the Kimura 2-parameter model. So here, it separates the two transition probabilities into transitions and transversion. You remember transitions is a change between pyrimidines or a change between purines, right? And a transversion is from one to the other. Okay? So here it's saying like, okay, any changes within the same time is equal, but different between purines and pyrimidines. But the base frequencies are the same. Okay? Then we have, so you can see we have different, I think by now we have over 200 and something models, because they make different assumptions. Okay? Ultimately, we have something we call GTR, General Time Reversible Model. Here it's saying all exchangeability parameters are free. So it means that I'm not making any assumptions. on these probabilities. They'll be determined on the data. And even with regard to the base frequencies, those are also free to change wherever. And even for this, there are a couple of complexities. Like, you can assume that you can, for example, say that, okay, are you going to calculate one rate across the whole tree, or you're only going to calculate a rate for a given cluster within the tree, and you expect that that will be different for another cluster in the tree. Okay? So, the message here is, there are different models with different assumptions.
But sir, there are three models, but now you give us a lot of.
Did I say there are three models?
Il y a beaucoup de paramètres à prendre en compte avant de construire une arme génétique.
So, James, maximum likelihood.
Oh, you see.
I think you have to tell us that before the construction of the tree, we have a lot of parameters to take off from.
Okay. Good question. I'll go back. No problem. Now, you see, even in the okay, I'll forget that. Now, let's say we are looking at different methods of constructing the tree.
Yes.
So here, on your left, these ones don't use any models at all. And on the right, these ones use some models. Okay? But even these ones that use models, there are different ways that those models are used. Okay? So we have these simple ones, the distance-based models. They use very simple models. And then we have these ones that use statistical approach. Here, there are also, there are different kinds of models. Here we are talking about how the probabilities are calculated. Okay? So we have maximum likelihood, which calculates likelihood based on the data. And then we have Bayesian statistics that typically you must have a prior. So you must have a prior assumption based on your data, and then you calculate that prior assumption times your likelihood to get your posterior outcome. So I'm trying to avoid going into the complexities, okay? But this is a general outlook of how the reconstruction methods work. Now, when you're in this section here, some of the models that we use to calculate the probabilities are, one, looking at the base frequencies, and two, probability transitions. Okay? So, do we assume that they are the same base frequencies? Do we assume that the transition probabilities are the same or different? Is that within the same, transitions or transversions, or are they all free? Okay? Tomorrow, I'll show you how we set these different models when running a tree. So don't worry. Okay? But I just need you to understand that, we have different methods. Some do not apply any models. Some do apply models. The ones that apply models, there are different ways in which those are implemented. You can have maximum likelihood, you can have Bayesian, or you could just have distance-based. Okay? The actual ways these are calculated requires a higher level of statistics. So I would want to keep this at this level still introductory. Then we can go to another session where now we can go into the different. Okay? Okay. Any questions? Do not be discouraged. We are good. C'est bon? Now, we've talked about different tools. So we have, for example, the parsimony. There are different tools that you can use, like POW, Nesquip, Phylip. I don't even know whether some of those are still maintained, but anyway. So here we want to find the tree with the minimum number of mutations between sequences. Very simple. Okay? Then we have neighbor joining. So these are often distance-based. So you estimate the genetic distance between sequences and cluster those into a tree that minimizes the genetic distance over the whole tree. So again, for the neighbor joining, some of these often don't employ any complex models. So they might just implement very, very simple models on how the distances are calculated. Okay? Then we have the maximum likelihood trees. I think I might have. Okay, let me see here. I think I might have a slide or something. Some of this is heavily statistical. So this is how you calculate the likelihood. So the probability of observing an outcome of data given a hypothesis. So here you're saying, what is the likelihood of the tree given your sequence? Okay? So essentially, you have this. Then you talk about, the likelihood parameters given the data. And here, in this case, the hypothesis is a tree, plus the branch lengths, and the data are the sequences. And then the Bayesian now is slightly more complex because you're saying you want to calculate the posterior, given your prior assumptions of the data, times the likelihood that you do calculate out of that data. Okay? But let's make things a bit simple, right? Arnold will tell you a lot of this knowledge is what you acquire over the years. So I'm not trying to teach it in one session, okay? Okay, so there are different tools that you can use for maximum likelihood and also for Bayesian analysis. For our own analysis here, I don't remember what epidemiology uses. Stephen, does it use IQ3?
Yes.
Okay, so epidemiology uses IQ3. Okay.
L'outil che epidemiology utilise per costruire la phylogenetic che c'est epidemiology.
Tomorrow, I think we'll run all these things and then we'll see practically. Okay. I know this is a lot. I understand. But we still have to learn it. Okay? So, remember, for us to build a tree is showing evolutionary relationships, right? So we expect to have what? We expect the sequences to have some mutations, right? And often what happens, we are collecting the samples at different time points, right? So there's something we call temporal signal. So pathogens are often sampled at different points in time. And we expect as time goes along, these pathogens are accumulating mutations, correct? Now, before building a tree, obviously you need to be sure that there is a temporal signal. Like there is sufficient information in your data set that you can use to build a tree. Otherwise, there is no point. Okay? Like, how many remember trying to build a tree at the beginning of SARS-CoV-2? At the beginning of the pandemic, you know, you could take the global data, put in your sequence from Kenya, and, you know, maybe you think like, my sequence came from Germany. The following day, you know, there are a few more sequences from somewhere. You put the same sequence and you're like, okay, it might have come from Uganda. Because at that point, there was very little temporal signal. Like there's very few mutations to clearly disaggregate your data. Okay? So, we want to have sufficient genetic change between sampling times to see some meaningful disaggregation in that data. So typically what we do is we make a plot of the divergence and time. And if there is temporal signal in your data set, you expect a line that goes like that, which shows that, you know, as you increase divergence, no, as I mean, you go through time, you're also increasing divergence.
Donc, il dit qu'effectivement, il faut qu'il y ait des signaux d'évolution dans le temps pour construire. L'exemple qu'il a donné, il a dit par exemple au début de SARS-CoV-2, de SARS-CoV-2, imaginez, vous avez une séquence. Puisqu'il n'y avait pas encore de signaux, tu mets dans un cours de séquence, tu peux te dire, voilà, ça vient pas de l'Ouganda par exemple. Mais demain, notre séquence, ça vient dans notre pays, parce qu'il n'y avait pas encore beaucoup de signaux de mutations qui peuvent effectivement renseigner sur l'évolution dans le temps. C'est pour cela que maintenant, avec la figure que vous avez, c'est la divergence sur les Y, et en bas, nous avons dans le temps, et on espère avoir, voilà, le type de figure que vous voyez sur le tableau. Plus vous avez divergence, plus ça évolue dans le temps, et également qui est dans l'axe des X.
Very well explained. So, sometimes we want to do this before we do a phylogenetic analysis. Okay? We want to see if there is temporal signal in the data. Okay? Now, this always works very well when you're working with RNA viruses.
Alors ça marche bien, surtout quand vous travaillez avec les virus à ARN.
Because they change too fast.
Parce que c'est eux qui changent, qui mutent rapidement.
So even when you look at a short time frame, you're likely to see something like that.
Même si vous observez tout cela dans un peu de temps, vous allez voir que la filature phylogénétique ressemble à ce que vous avez sur l'image.
For DNA viruses, they change very slowly.
Et pour les virus à ADN, ils mutent voilà, très difficilement à une vitesse qui est très faible.
So you might only see this after a very long time.
Pour avoir une figure comme ça demande quand même beaucoup de temps.
Okay? So, again, I'm giving you all the principles, but it does not mean that it's always applicable. There are times like, okay.
Donc, je vous donne les principes, mais ça ne veut pas dire que toujours ils sont applicables.
Okay? So, like you could say, if there's a linear trend, good. If there's no trend at all, so this line, the regression line. I don't know, you learned regression line in somewhere in campus. If it's flat, it means there's no trend at all. But sometimes you might actually have a line that goes like this, which simply means like, you know, as you go through time, the divergence is getting lost. It just simply tells you there's something wrong with the data.
Donc ici, il faut avoir quand même une de régression qui vous indique sur la tendance. Et des fois, ça peut être oblique comme ça. Et ça veut dire que quand même, il y a un problème avec vos données. Ça veut dire que plus vous avancez dans le temps, plus la divergence est perdue. Il n'y a plus de divergence.
Sometimes, actually you might just get something like that. And it could simply be because you've included a very old sequence. There is something wrong.
Donc, des fois, ça peut avoir le sens contraire, par exemple, comme ça oblique, et ça veut dire que vous avez utilisé quand même une séquence qui est très ancienne, et du coup que ça les données ne sont plus, on a plus de sens.
But then again, even when the data looks like this, it does not mean it's correct.
Mais même si ça donne cette tendance-là, ça ne veut pas dire toujours que c'est quand même de bonnes données.
Okay. So analysis is complex. So you should always look at data from different angles.
Donc,
Alright?
c'est tellement complexe qu'il faut toujours avoir un œil critique et avoir les informations des angles différents.
James,
Alright.
do you have somewhere you can look like a recombinant in this? Because it seems always
Or I don't have one for recombinants, but I think I might have. I'll go back to this slide set. Maybe we'll make. Okay. So there's a tool that we call Tempest, where you can upload your tree, and if they have dates, you can try check for temporal signal. Okay?
Donc il y a un outil qu'on utilise, voilà, où si vous avez des données avec des dates, vous mettez dedans et ça vous montre quand même.
Yeah. So, I mean, I could show both. So you see something like this. So here we have divergence, there we have time. Now, for the most part, because when you're selecting your samples, they should be maybe part of an outbreak, they should be the same clade, things like that, right? So you expect a lot of the points to be clustering around the regression line.
Donc des fois, vous avez des séquences qui proviennent de la même épidémie, ou bien du même clade, et là, vous avez quand même qu'ils se regroupent autour d'un même cluster.
But what if you see something like this?
Mais qu'est-ce qui se passe quand vous voyez par exemple une séquence qui est de l'autre côté en haut, loin du cluster?
Man, it's just very different, right? It's very different. So when you see things like that, things that we call outliers, right? I think the initial reaction is you need to remove that. Okay? Because it will, I don't know what the English for, skew your data. You're the statistician. So, I mean, it changes how your data looks. Okay? And even the tree will be slightly, you'll often see it with that. I don't know if I have the tree here. Oh yeah. So you can see that here most of the branch lengths you can see how they look. But the outlier you can see how long it is. Like it's very different. The branch length is very long, right? And that's why when we're seeing things like this, we need to remove such an outlier from our data set.
Donc, des fois, quand vous avez une séquence, par exemple, un échantillon qui est loin des autres, normalement, ça a tendance de fausser vos résultats. Et qu'est-ce qu'il faut faire? C'est mieux de l'enlever pour continuer avec ceux qui sont quand même dans le bon sens. Et ce qu'il a montré, the next one, this one, et même,
Oh, you want the tree?
il y a des trucs. Et voilà maintenant, peut-être qu'ils sont comme ça, mais l'autre, vous voyez la distance. Il y va très loin par rapport aux autres. Donc c'est là où il faut quand même l'enlever pour continuer avec ceux qui, voilà, qui sont dans le même cluster.
So checking temporal signal has two functions. One, you want to ensure that you have sufficient mutations in your data set to generate the tree, and two, to remove any outliers.
Donc c'est très important et ça a deux fonctions. D'abord, c'est de voir qu'il y a assez de données, assez de marqueurs pour continuer, mais aussi c'est pour voir s'il y a moyen d'enlever ce qui pourrait fausser le résultat.
Good? Yes,
What if the outlier is no one mutation?
that's a good question. So you're saying that what if the outlier is
La question
just
qui
something
pose,
new?
c'est de savoir qu'est-ce qui se passe si jamais la souche là qu'on veut éliminer, c'était une nouvelle mutation.
That's a good question. So obviously, what you want to do at that point is to go back and ask yourself, like, okay, how does the assembly look like? Do we have, for example, enough coverage? Peter showed you how to like get the BAM file and view it on IGV. The mutations that you're seeing that makes this different from the others, do they have sufficient coverage? Was it good quality? Did the reads have good quality values? Okay? So you can go back and try and determine, is this real or it's not real, right? Because I agree with this point. You might have discovered something, so you also just don't want to throw it away, okay? But what you're saying is you must be very suspicious of something that is different. Yes, Steve.
Just question for just now. You say that our supposed to wear this will apply. Looking for a temporal signal, like whereby it would be difficult for us to apply such a thing.
When you don't have,
Like you, I earlier had said that there are some scenarios we would have to see that doing a temporal signal to probably the mutation rates are not high. I don't recall how.
oh yes. So one of the examples I gave was earlier in the epidemic. Earlier in the epidemic, you have a few sequences and you expect very few mutations. And the reason you expect few mutations is that you're still not, the population has not developed like strong immunity against the pathogen. So you're not going to say that I'm not going to analyze that data because there is very little temporal signal. Of course, you have to analyze it and see how the pathogen is evolving. It's just that at that point, you are aware that, okay, these are the circumstances, okay? But if you're well into the epidemic, you expect some temporal signal. So if that is not happening, again, you have to like, is it something about the biology? Is it something about my sampling? Is it something about my choice of sequences? See what I mean? So also the context matters. Right?
Now the other challenge we have, now usually doing the collection of data. So how true is that? Sometimes the bits of collection that are being provided, someone using a phone, how full is this data?
That's a very valid point. And it's correct that if you have the wrong collection date, actually you'll see it as an outlier in that plot. And you see sometimes the mistake people make, a sample was collected last year, 2023, but you've sequenced it this year in October. So somebody, instead of putting the collection date, they put the sequences date. Often that will show as an outlier on this plot because the divergence will not match the expected date. Okay? So that's also how you can spot such errors.
Yeah, so something else I've seen is it depends with your tool of some of metadata collection. So there are people who put year, month, day, and then the people who put year, day, month. Right? So if you get something like 01/07/2024, how I'll interpret it is very different. Is it January? Is it July?
Okay, now we go into the discussion of how Americans write their dates
Yes.
versus
The UK.
how the rest of the world.
How the rest of
Actually, it's America versus the rest of the world. So,
yeah. Yeah.
yeah, that's a very valid point.
And if you use time tree, it tends to distort it. So a better way is to use your dates as decimals. Then that's
usually very accurate. Yeah, that's a very good point. You'll always see it in history, like it's good to spot any outliers. But then again, you see, you can investigate whether those are real, would they be typographical errors, somebody just wrote the wrong date, or it's just bad quality sequence. Okay? Okie dokes. So, where do you want to stop before T? T is at 4, right?
No, anytime between 3:30 and 4.
Okay.
So
All
just to add on to that point where you're saying when you're collecting data, so when the Wuhan virus was first discovered, they actually didn't release that data until like more than a year later. So that in that time they were collecting data from around the world, and a lot of the data was actually not stored properly. So even when they sequenced the data, you could see like there were discrepancies. And later on, when they actually released the data collected from Wuhan, they actually, I think James showed you those 4 different phylogenetic trees, right? They're actually from 4 different genes. So you actually need a large enough sample pool to determine the statistical average almost. So you can't just take one sample, say, this is my final result. You actually need to take a number of samples over a long period of time, and that will give you sort of an aggregate value and eliminate as much error as possible.
Yeah, very right. So, yeah. Any questions? They are buying question. So, one of the things we talked about was rooting your tree. So, phylogenetic trees are either rooted or unrooted. Okay? If you do have a rooted tree, you have some sense of directionality. You want to say that, these are the parents, these are the descendants. But sometimes you're not interested in rooting a tree. You're just interested in the diversity, And often people would use an unrooted tree for that. So, to root an unrooted tree, so one, you can root by an outgroup, okay? Which means it's a sequence that is closely related, but not clustering within your sample. So, for example, there let's assume that your samples say A, B, C, D, and E. Then you use F as an outgroup to root that tree. Okay? The other rooting that you might do is midpoint rooting. So midpoint takes the 2 most divergent sequences and then puts a root somewhere in between. It's often used to just give your tree some sort of balance on how it looks, right? But remember, rooting by an outgroup is much better for giving that sense of directionality. And we've seen that an outgroup need not to belong to the in-group, so it should not cluster within A, B, C, D, and E. It should be the most distantly related to that taxa, but not be too distantly related. And of course, it should be homologous, which means, they should be either the same species, for example, or, similar clades or things like that, right? So you cannot be analyzing clade 1B sequences and you take a clade 2B as an outcome, right? You'd probably take a clade 1A is better than using a clade 2A or 2B as an output for clade 1B for the genetic tree.
Bon, là c'est quand on a un phylogenétique, comment on va dire en français, voilà, pour mettre la racine. Voilà, pour mettre la racine, il y a 2 façons de le faire. Soit il y a ce qu'on appelle root by outgroup, ça veut dire quoi? Vous allez utiliser une séquence qui n'est pas forcément reliée à vos séquences. Ça qui va pas se mettre dans le même cluster que vos séquences. Souvent, c'est une souche qui est relativement associée à ce que vous voulez analyser. Ou bien des fois, il y a ce qu'on appelle le midpoint routing. Vous prenez deux souches qui sont diamétralement opposées, si on peut dire comme ça, et la machine va essayer de faire au milieu pour donner la racine. Mais le meilleur, c'est le root by outgroup en utilisant une séquence qui est un peu différente de celle que vous utilisez. L'exemple qu'il a donné, c'est par exemple avec le Mpox. Par exemple, vous analysez Mpox clade 1B, vous n'allez pas utiliser le clade 2A ou 2B pour enraciner la phylogénie. Par contre, vous allez utiliser par exemple le clade 1A qui n'est pas le 1B, mais qui est proche de 1B pour donner la racine à votre arbre. Voilà.
Perfect. I think tomorrow we'll go through an exercise where we'll do this, so again, don't worry. So this is how an unrooted tree would look like. So remember, we had this tree where we had the root F. When we remove the rooting at F, now we have an unrooted tree, and here we are only focusing on the relationships. We are not thinking about the direction. Okay?
Il y a des fois où on n'a pas besoin d'enraciner la phylogénie. Là, c'est juste pour voir la diversité. On n'a pas besoin de voir la diversité, c'est juste pour voir la divergence au niveau de nos séquences.
Okay. Probably the second last thing before we go for break is the concept of bootstrapping. So, when you generate a tree, often we ask how robust or how reliable is your tree. Okay? So, bootstrapping is a statistical technique that uses random resampling to determine sampling error. Okay? So it gives an idea of reliability. So usually we think about a bootstrap more than 70%, showing that that cluster is reliable. I'll give an example before you translate. So let's say this is a phylogenetic tree. At this point, we have a bootstrap of 90. And on this node here, we have a bootstrap of 65. So, and on the next slide, I'll show you how we do the resampling. So we are saying that, let's say we did, this is out of 100%. So 65% of the time, this node clustered together to the exclusion of whatever is on the left. 65% of the time. Very important. 65% of the time, this node clustered together to the exclusion of whatever is on the left. Okay? This node says 90% of the time, this node clustered together to the exclusion of whatever is on the left. And if you think about this bootstrap value, which one do you have more confidence in? 90 or 65? 90. Okay? So before you make any strong conclusions about a given cluster of sequences, we often look at the bootstrap value. The higher it is, the more confidence we have about that cluster. So for example, let's say this one is a cluster of sequences from Uganda. If you have a bootstrap of 90, we can say like, oh yeah, Uganda sequences cluster together always. And we have very high confidence, right? But if the bootstrap value is like 65, you're like, you see, like 35% of the time, this Uganda sequences cluster somewhere else, right? So you're like, okay, you're not too sure. Like 65 is not below 50, but still. You want to translate?
C'est une technique utilisée en statistique pour donner de la confiance lorsqu'on fait c'est pour l'efficacité de comment est-ce que je peux dire je suis confiant qu'un groupe de séquences forme un même cluster. Donc, cela, on va regarder ce qu'on appelle la valeur du bootstrap. Plus la valeur du bootstrap est élevée, ça veut dire que dans la majeure partie, on a 90%, ça veut dire que dans la majeure partie des séquences qui sont dans cette zone, on a tendance à se retrouver au même ordre. Si on trouve une valeur du bootstrap autour de 65%, c'est ensemble, 5%. Et généralement, la valeur du bootstrap devient threshold à 50 c'est 60%. Et dans tous les cas, ce qu'on va aller chercher le plus, c'est les valeurs ici. Plus la valeur du bootstrap est élevée, plus on est beaucoup plus confiant que ces échantillons font de bootstrap.
So, here, we'll show you how we calculate the bootstrap on a tree. Say you have an alignment, okay, of sequences A, B, and C, correct? And it has positions 1, 2, 3, 4, okay? So first, we build a tree based on that alignment, okay? Then we have a tree here. And the next thing we do is we randomly choose. So let's say we have an alignment that has 4 positions, right? So in the next round, we randomly select 4 positions from this alignment. So we could take this twice, this position 1. We could take it twice, and take this position once, and this position once. So we have an alignment of 4 positions. That's what we want. So in this replicate, we've ignored this position. Then we build a tree. Okay? Then that's how the topology is. Then we do again replicate number 3 or 2. We build a tree based on, again, a random selection of different sites. So you can do this, typically people do this for like a bootstrap of 1,000. So you do this 1,000 times. Now once you've done this 1,000 times, you ask yourself, how many times out of the 1,000 were B and C seen clustering together? You assign a bootstrap value. Okay? How many times were A and C seen together? How many times were B and A seen together? And then, you get a summarized tree that summarizes all these probability estimates. Okay? And that's how you end up with a value like this and this. Okay? So if you look at here, you can see that in one of those replicates, A and B cluster together, right? Here, B and C cluster together, B and C together, B and C together. But there's one replicate where A and B cluster together. So if we were even to just use like, let's say 1, 2, 3, 4, okay? We can say that B and C clustered 3 out of 4. So that's around 3 out of 4 is like what? 75%. Okay? But there's a 25% probability that it clustered with A. Okay? So this gives us that measure of reliability in the individual clusters. Because that's often very, very important. Like, it is expected that when you build a tree, there are some sections of the tree that will have low bootstrap values. That's fine. Okay? What is important is, for that node, what kind of conclusions do you want to make about it? Okay? I do not expect that my tree will have high bootstrap values throughout the tree. No. But I want the part that I'm interested in making conclusions about has good reliability. Okay? All right. I think we can take a break, and then we come back and see how we can interpret a tree.